So today's Let's Talk MPTs webinar discussion is going to focus on toxicity study considerations for MPTs and HIV prevention R&D with two leading experts in this area, Dr. Tom Munch and Dr. Miles Brennan. As I am sure you know, this is a really critical and complex topic, and it's all the more so when we're thinking about combining more than one active ingredient. While 60 minutes can only provide us with so much time to cover these topics, we aim to highlight key issues and also connect leaders working across the disciplines to further stimulate innovation and collaboration. So I think everyone is joining. So we have, as I said, 50 people registered, if not more. So that's really great. For those who are new to the MPT field, I wanted to just remind everyone what MPTs are. They are products designed to simultaneously address two or more indications, namely HIV, other sexually transmitted infections, and/or unintended pregnancy. They could be one-stop shop products that revolutionize sexual and reproductive health by incorporating prevention of HIV or other STIs into contraceptives, which are less stigmatized. They can also reduce the burden of multiple clinic visits, which has become more of a challenge due to COVID, and which we can each all likely relate to at this point. There are an array of about 24 MPTs in development with active funding. MPT candidates include various combinations of contraception and prevention from HIV or other STIs. While the majority of the MPT candidates are in early stages of development, some are in clinical stages. And you can check out the MPT product database at mpts101.org. If you know of other MPTs that are not included in the database, please reach out to us. We're constantly updating the database and doing our annual formal update actually right now. So the Let's Talk MPTs discussion series was actually launched to engage experts from diverse disciplines from across the globe to address MPT and HIV prevention, R&D priorities and gaps. And the topics that we are including in this series have been informed by our MPT partners, including responses from a stakeholder survey that we sent out about 10 months ago. And as with past discussions in this series, we're really looking forward to an engaged and lively discussion today. Past topics have included the role of cervical vaginal microbiota on contraception, HIV prevention and MPTs, and the integration of socio-behavioral market research. And we have several more webinars coming up. All the webinars and recordings are available on the website. So today we're really excited to welcome our featured discussants. Dr. Tom Munch is a clinical infectious disease specialist and an expert in contraceptive and vaginal microbicide development and formulation. He is currently the Chief Medical Officer for MAP Biopharmaceutical and Vice President of Clinical Development at UConn. Miles Brennan has worked in a number of fields, and a common theme of his work has been the genetic engineering in various systems to address important questions. And for the past 6 years, he's been working on various aspects of monoclonal antibodies for infectious disease. So Tom is going to be giving the formal presentation, and then we'll be opening it up to question and answer, and Miles will be participating and with a lot of that from regulatory perspectives as well. So Tom, hand it over to you.
Great, thank you, Bethany. I just want to thank you and Susanna for organizing the webinar, and for those who are attending, and thank Cami and IMPT for their important work in advancing these preventive products. Next slide. I'm going to go over in the webinar these topics. the need for special vigilance for toxicity issues in prevention products, particularly. The goals of toxicology studies, which may be a little broader than you might at first think. Implication of product types for what kind of toxicity testing is needed. Some examples of non-clinical toxicity programs or plans. And then finally, resources for preparing a plan for the development of individual products. Next slide. So first to this issue of the need for special vigilance in preventive products. These products are intended for use in a healthy population. And that's quite distinct from therapeutic products where the degree of side effects that it would be tolerated is greater. Here you're starting off with somebody who's healthy and you're trying to keep them healthy, but there's a greater need for caution in terms of inducing toxicities and a lesser degree of acceptability of toxicity. So this toxicity topic is particularly important for MPT type products and any kind of prevention products. And particularly because these things might affect people's reproductive health, which is always a critical concern to people who might be using these products. Next slide. Here's a slide that kind of covers both what we're going to concentrate on mostly in the circle, which are the studies that are done in advance of clinical trials, but also things that are done after clinical trials begin and are done in parallel with the clinical trial. And that's the genesis of this word non-clinical instead of just preclinical. It used to be called preclinical. And in fact, the EMEA still uses both of those terms interchangeably. The FDA uses non-clinical, and this is the reason. Some of these studies are done after clinical trials have already begun. And they really span a wide range of things, and we'll talk very briefly about some of them on the right, but we're going to concentrate mostly on the studies that you need in order to get into your initial clinical studies with this kind of a product. Next slide. So even before getting into the studies I mentioned on that slide, the previous slide, there are some very early evaluations that will likely be done. And depending on where a product is in its development, these things very likely have already been done. But for people who are just starting out with a new product, a new idea, these things are things you'll be doing very early on. You'll do preliminary studies to determine if your product is likely to be safe, meaning in vitro studies of cytotoxicity. Those can be done in monolayers and nowadays can also be done in three-dimensional cultures that if this is a topical, that would be highly pertinent to their use in the vagina. And the kind of things that are looked at there are viability, barrier function, trans-epithelial resistance, and also not on the slide, but induction of inflammatory responses. And these kind of results are compared with in vitro effectiveness and to try to establish a therapeutic ratio. In other words, how much lower concentration compared to the toxic concentration is still therapeutically effective. And really, ideally, those should be ordered of magnitude different. The next thing to mention is something that is important for people who do device studies. If your product is a combination product that included a device, and that is to be aware of the existence of a pathway to get some early information that might inform your design. And these are called non-significant device studies. And there's a link there that gives more information about them and how that can be used. These can be used at a point where you're trying to design what your device should look like and what the device without an active in it, what its safety and acceptability and other parameters may be. And so you may want to avail yourself of that. And that can be done at a point where you have not yet approached the FDA, and the regulation of whether you're safe to proceed with those kind of studies rests in the hands of the IRB that you will be using for those studies. And finally, another resource available to you is something that's called an exploratory IND, where you do what are called exploratory studies at a point where you may have less non-clinical data than a full IND, and you may be able to get into the clinic to answer some important questions. And that can be important as you're deciding on feasibility, trying to determine effect size, exploring whether the mechanism of action that you think you may have can be confirmed in a human study, and just generally to do proof of concept work that might enable you to get more funding or more interest from other parties to bring your product forward. So that's something to keep in mind, and the link there gives you information about how that's done. Next slide. Well, I want to talk about a number of ways to think about product formats and product types, because they influence what kind of studies need to be done in order to move forward to clinical studies. And the big two divisions are systemic versus topical. In systemic, there's really a wide range of dosage formats, some of which are open for sustained release, and others that might even be used for on-demand use or precoital use. And likewise, topical, there can be on-demand precoital type products, gels, films for rectal protection, rectal douche. And then there, also in topical agents, you can have long-acting agents, and intravaginal rings and long-acting films are examples of those kind of topical formats. Next slide. So looking at that big division of topical versus systemics, and thinking a bit about the pro and con, the big advantage of topical products is reduced interactions with the internal body systems, at least in terms of toxicity issues. This is a big advantage. They also have and have been used with on-demand precoital format, which is a product format that's needed, although, as I talk about next, has some limitations as well. Limitations of topical agents are that there can be local toxicities right at the point of entry for STIs and HIV. And this actually can both apply to the active pharmaceutical ingredient, the API, but also even the excipients of your product might reduce epithelial barrier function. And that function is very important. There's a considerable barrier to the establishment of these infections. And if you lower that barrier, you might reverse the benefits that you're hoping to have. So that's very important to keep track of. There can be interactions with the microbiome of your product when it's applied topically, and that can have adverse effects on barrier function or induction of inflammation that may make HIV in particular more easily established. But the downside also is that, at least if these are paracoidal dosing formats, then there can be challenges for adherence. On the systemic side, an important advantage is you will not likely cause these cervical vaginal epithelial perturbations. And further advantages, there are several of these formats that look very good for giving prolonged action and therefore helping with adherence to the regimens. And on the negative side, converse to topical, you're exposing all body systems. As mentioned, the tolerance for side effects is low, so the more systems that are involved, the more that becomes an issue and things that have to be looked at. And then the related thing is interaction with other drugs, and many people are on many drugs, and your drug might end up being a victim of another drug's presence or the perpetrator of an adverse interaction with other drug therapy. So that's kind of the pro and con of two different categories. Next slide, please. And another way to think of categories is what kind of drug is it? A small molecule drug, a much larger biological, like a monoclonal antibody or a messenger RNA approach. Interestingly and kind of paradoxically, these, at least the monoclonal antibodies and probably the mRNA, if it's going to be inducing expression of monoclonal antibodies, are regulated by the drug center rather than the biological center. and that has implications in terms of what some of the requirements are. And then another category is sustained release products like vaginal rings or systemically released drugs from implants. Another category could be a combination product in the sense of more than one active agent, maybe no device involved, but multiple agents. And obviously for MPTs, that's very likely to be generally the situation since you're attacking multiple targets at one time. Next slide. So getting into some of the toxicology studies that are needed, I think we primarily think of this as a gateway to get into doing clinical studies, and that's absolutely correct. But there are other goals that might not be quite so obvious, that other things that these early toxicology studies help you with. One is to estimate a safe starting dose for the first in human phase 1 study. And to that end, you attempt to establish a no observed adverse effect level, or NOEL. And this doesn't necessarily mean no effect, but no adverse effect. Some minor changes that are not judged adverse might be present, and that might still qualify as a dose that is a NOEL level. And secondly, on the other end of the dosing spectrum is to establish a maximum tolerated dose. And that informs the upper dose level for your phase 1. And in fact, it's important to try to do that. And in a way, finding some toxicity at some upper level is actually quite advantageous, a little bit paradoxical, but it's very helpful to have established that. And you really are obliged to try to do that. Now, sometimes that's not possible, and hopefully the agent may be so non-toxic that it's not possible. But what you have to demonstrate there is that you have made a big effort to determine that, either by achieving exposure saturation, for example, an oral drug that eventually you don't absorb anymore and the blood levels don't go up, or using a maximal feasible dose, the highest concentration you can achieve, and the limited volume that can be placed if it's an intravaginal study, etc. So you've gone as high as you can go. And then another a way to meet this requirement is to achieve a very substantial exposure market, maybe a margin, maybe on the order of 50-fold above what you're expecting to use clinically. And related to that, second bullet is that what this does is it helps you identify specific safety endpoints that you will be monitoring closely in clinical trials. So you have to know what could go wrong with this drug and then be on the lookout for it once you enter clinical trials. Next slide. So, and then as was seen in that earlier slide, and I'll show you again later, there are later toxicology studies that are a little bit beyond the scope of what we're going to discuss this morning, where you are perhaps trying to identify the consequences of chronic exposures, not just the acute study that you do in order to begin your clinical trial, but what would happen over the long term, since these products are expected to, in many cases, be used over the long term. And secondly, to predict risks that you really can't very directly assess in humans, either because of practicalities or ethical issues, such as carcinogenicity, reproductive performance, and developmental toxicology. Next slide. And there are some other general principles of the toxicology program design. One is that you will generally include animals that are sacrificed later after exposure than the main group. And there the goal is to see if toxicities are demonstrated in the main group, whether they have resolved after the drug is withdrawn. And so the duration of that recovery phase will depend on the half-life of the drug and other factors like that. But you'll be looking to see whether any toxicity that's developed has been reversible. Then toxicokinetics, which is a little kind of a fancy word, but just it's pharmacokinetics in a toxicology study. And another way to distinguish it is in this kind of a study, you're going to be looking at doses that may be much higher than you're planning to use in the clinic. And So it's distinct from the pharmacokinetics that might inform or might be studied in the clinical study itself. These can be studied in animals and can't be studied in the clinical study. And you may be able to make correlations between those high exposures and whatever toxicity you see, and then try to prove eventually that the exposure levels you're seeing with an effective dose in the human studies don't need to go that high. So that's the goal there. And finally, that the duration of these early studies have to support the length of the planned clinical study. And those clinical studies get longer over time, and therefore additional studies need to be made with different lengths. Next slide. So here, in fact, are the guidance on what kind of study length is needed in order to get into a study of up to 2 weeks, which would be kind of a typical first-in-human study. You need a 2-week study in rodents or non-rodent species. And again, I think I may have failed to mention in one of the other slides on principles, in many cases, with a few exceptions, 2 species are going to be required for your toxicology studies. So 2 weeks to support that kind of a study. There are some exceptions, again, and monoclonal antibodies, sometimes they're studied a slightly shorter period of time, as I'll show you. Then for later studies in the clinic, you need to use a length in the animals that will match the length of the clinical trial. And then with the idea in mind that these products eventually would be used over really quite a long time, possibly even decades, the length of study in rodents is 6 months, and for non-rodent species, 9 months. In other words, you're not required to study the product for decades. You study what's practical, and that's considered to be 6 and 9 months. And the guidance on this is listed in the link on the bottom of the slide. Next slide, please. Well, the different kinds of things that would come up with HIV prevention or MPT type products really are quite varied, and programs are distinct depending on what it is you're studying. You might be studying a small molecule systemically or a combination of small molecules. You might be studying small molecule or molecules topically. You might be using a monoclonal antibody either topically or systemically. And you may have a combination product such as a vaginal ring or an implant, and they have distinct requirements. I'm going to concentrate in the rest of the talk on the latter 3. Next slide, please. So the study of a topical small molecule, typically you'd need a 2-week good laboratory practice rat toxicology study. And that would include safety bloods, behavioral observations, other clinical observations, and then at sacrifice, gross pathology and histology. You would be gathering pharmacokinetic, toxicokinetic data, mostly blood levels of your drug. There would be a recovery phase. For most of these studies in the field we're talking about, you would use a single sex, females. That's contrary to broader development of a drug where you'd have groups of both males and groups of females. In a second, you'd be required to do a second species that would be a non-rodent, dogs, sheep, monkeys. And for topical vaginal drugs, that is most often met with a rabbit vaginal irritation study with additional endpoints such as, potentially such as the histology that goes beyond the irritation questions that the typical rabbit vaginal irritation study would address. And then if your small molecule is absorbed more than to trace amounts, you may have to do the various kinds of studies required of a systemic drug, including absorption, distribution, metabolism, excretion, and really a fairly extensive group of other studies, because this is, even though applied topically, may in fact be a systemic drug. Next slide, please. So next, talking about monoclonal antibodies. These have potential for IPT applications and anti-HIV applications. They can be combined in order to cover different targets, or in the case of HIV, to cover different HIV strains with different susceptibilities to the monoclonal. And they have certain things wherein they're easier than other drugs. Things that are not needed. The ADME type studies are not required. It's sort of understood how these molecules are processed in the body, and when they have a different FAB, they don't change that very much, so those studies are generally waived. Likewise, genotoxicity, reprotox, development tox, carcinogenicity studies are also generally not required. Secondly, a single species is often acceptable, at least if the drug would be an anti-pathogen monoclonal, which would be the case in the kind of products we're talking about here. Something new that's required, that's not required for other things, is an immunogenicity assessment, i.e., does your antibody cause the development of anti-drug antibodies, antibodies directed against your monoclonal? That has to be assessed in clinical studies, but it also needs to be assessed in animal toxicology studies. And there, depending on the lack of the study, it's almost universally going to happen because you're putting a human antibody into a non-human species. But the issue here is to determine whether, is to prove that this anti-drug antibody response doesn't limit the exposure by neutralizing the antibody. And so that's why ADA testing is required in the animal studies. Another additional test that needs to be done in monoclonals is, one of the reasons they generally have low toxicity is because they're highly specific, but they can have off-target binding, and that's investigated by studies that are called tissue cross-reactivity studies. And here, human tissues are exposed to the antibody and determine whether the antibody binds to any cells or tissues. And at the same time, you look at the species that you think you may be doing your toxicology studies in. And if you see off-target binding, that will determine which species are appropriate for toxicology studies. In other words, you'd want to choose a species wherein that same off-target binding was present. So you might have been hoping to do a rodent study, a rat study, but if you don't see that off-target binding in the rat, you may have to move that to be a non-human primate study. Next slide. So here's an example of a study for a monoclonal antibody. All of the animals were female in this study. It was intravaginal exposure. There was a control and 2 active levels. Again, there's a difference in monoclonals. In most small molecule drugs, 3 different levels of low, medium, and high are required. And you'll see that there are groups in the main group, which is sacrificed at a given time right after the end of dosing. And then after delay, a recovery period, additional animal sacrifice at that point in order to see whether any toxicity observed in the acute animals has resolved by the recovery day. Next slide. So here's an example of what kind of testing requirements and toxicology studies would be done for a drug plus device. First of all, a 2-week GLP rat toxicology study, same sort of things mentioned previously as the endpoints. Recovery phase required, females only for the kind of products we're talking about. But then in addition, there would be a biocompatibility study. And I'll talk more about that in the next slide, but this is because of the device component of this product, that kind of study is required by the device group, which will also be weighing in on your FDA evaluation. And then again, if there is more than trace systemic bioavailability, other studies will be required. Next slide. So here is the from the device group, which is referenced up at the link at the top. We would be talking about mucosal contact and a duration typically of 30 days or less. And the circled items are things that would be required to be done. There are various contractors who are used to doing this kind of work, and you go to them and they would work with you on this matrix to find out what your product requires, and they would do the testing that's required. Next slide. And then here, if your product is a drug plus a device with a vaginal ring, you'd use a 2-week rat study as described earlier. You would likely also need to do a study in sheep because in the rat study, you wouldn't be able to use the ring, you'd be just using the API. And then you'd be required to do a biocompatibility study. Next slide. So then a few final issues. It's worth thinking about the excipients in your product. There is an FDA list of inactive ingredients, and you should check that as you're designing your product because it's helpful if your excipients are on this list in the dosage form, i.e. vaginal, for example, if it's a topical, and in the quantity used in other approved products. That gives you a little bit of a pass for special scrutiny for those excipients. Excipients, not only APIs, but excipients can cause toxicities, particularly in topical agents, and particular caution on humectants or osmotic agents. Products that have a very high osmotic strength look like they are capable of damaging the epithelium, possibly even the microbiome. And then also I'd caution you on high concentrations of EDTA. That's often used as part of the antimicrobial package, but it can reduce barrier function of the epithelium. And then another interesting question is what should the control be for toxicity testing and clinical testing? And your choices there are the vehicle control, which is in many ways has lots to recommend. It's a super good scientific experiment. You're comparing the vehicle with the vehicle plus the active, so you're looking at changing one variable. But on the other hand, for microbicide studies, there's been a move for topical things like gels to what I'm calling a certifiably bland control, something you have got great confidence would neither harm nor block infection, so that you can really see the effect of your product, and that's something to consider. Next slide. So showing this one more time to say, we're concentrating on the non-clinical studies required to get into your first clinical studies. There are other studies that may be required in parallel with your clinical studies. These are longer, more expensive, more time consuming. And fortunately, they don't need to be addressed right away as you get experience to find out whether your product looks like it's got the ability to go forward. Next slide, I think is the last slide. Yeah, so in summary, thinking about how you prepare a plan for your product. I went through all the different forms of products and product formats and dosing regimens because they influence what's required for your toxicity program. And so there's a real need for a customization and consultation with the FDA and negotiation with the FDA in terms of making a case for a program of preclinical testing that you want to do. There are guidelines that are very helpful, and those are referenced in the IMPT materials that go along with this talk. And you're going to want to engage appropriate consultants for complicated products and systemic products. Toxicologists may be useful to engage. Certainly regulatory affairs at an early stage for guidance. And then jumping to the last bullet for a moment, the best consultant and the free consultant is the FDA itself, and that's available to you in what's called a pre-IND package, where you give information on your drug and you propose a plan, and they comment on it. And so you're making a concrete proposal and seeking their comment. And then jumping back up one bullet to just mention, there is a potential NIAID resource that I think has not been yet heavily utilized by people in our field, but that gives you resources and the ability to do studies for IND enabling studies, and that's called the Comprehensive Resource for HIV Microbicide and Biomedical Prevention, or CRIMP. And I've got a link there, and you can look into that and see whether that might be a resource that you could use. So I'll end my talk there, and thank you for joining the webinar, and turn it back to Bethany for handling the discussion.
Tom, thank you. That was really fantastic. Really, really appreciate all the work that you put into preparing this talk and all the resources that come along with it. We're now going to open it up for question and answers with Miles and Tom. And so if you have questions, you can submit them into the chat. We also would welcome, we do want to facilitate, sort of community through this platform too. So you can unmute yourselves and ask questions either by raising your actual hand or your virtual hand, and then you can announce yourself. I think, so I want to actually hand it over now to Joe Romano, who I think many of you know, and he has a question for Miles and Tom. Joe.
Thanks, Bethany. And Tom, thanks for that presentation. Incredibly comprehensive and informative. I have a couple of questions based on what I heard today. First, when contemplating toxicity studies for products that involve multiple APIs or drugs, are there any relevant considerations, particularly on the in vitro side, that would be important to conduct or complete in order to inform design on the in vivo side of a toxicity study? For example, in vitro, are there drug-drug interactions? Can you assess the metabolite portfolio from simple cell culture type assessments that you may want to take into account in how you design a in vivo study in animals?
Yeah, thanks, Joe. Miles, do you want to take a crack at that?
Oh, you're on mute.
Sorry, I'm technically challenged. No, I mean the in vitro experiments that you would do, especially with products with multiple APIs, these get to be really important because you're worrying about the ability of one drug to interfere with either the mechanism of action or the toxicology of the other drug. And so this becomes a real big issue. And there have been cases where, and especially with small molecule drugs, where you can imagine that the detox pathway of one will interfere with the detox pathway of the other. And all of a sudden your PK has gotten much different than it would in with the single agent.
So to go back to a point, Tom, you made, well, thanks for that, Miles. And to a point you made about controls, if you're dealing with products that have more than one drug, you need a vehicle only, presumably, as well as a formulation that contains just one of the drugs, and the other, the test article being the one that contains two drugs. Is that going up if you got something with three drugs? So you'd have to have these partitions of controls so that you're understanding if drugs together have issues or not.
Right. I do think those kind of studies are likely to be required. This issue of combination products is actually a very complicated one and a bit beyond the scope of what we can get into today. And I believe there either has been or is going to be some specific attention to this in future webinars. I mean, the initial FDA approach to this was very restrictive. I mean, prove A works, prove B works, prove A plus B still works and is better than either A or B. I mean, really hard to do studies, even including clinical efficacy. And they did that at an earlier time relating to a different goal of preventing people from just throwing two things together and saying, here's a nice combination product. But for, what we're addressing here is even HIV by itself, or certainly, MPTs beyond HIV only, combinations are almost certainly going to be required in every case. And they're required, throughout, not just to show, well, we're better against this one agent, but because our target is multiple things that can happen during pregnancy that we want to prevent. And
Right.
so I think they've come some distance there, but I'm not an expert on that. And I really encourage IMPT to have more discussion of that. I think that's, something that can hold us back if we don't get a plan that includes agency buy-in in order to move these products forward efficiently.
And I would just add to that, just from the standpoint of, say, antiviral antibodies, that the agency has become very accepting of cocktails, and actually in some cases demanding or expecting cocktails where the drug targets may be overlapping to avoid, let's say with HIV, to avoid escape. Tom?
Yeah, I think that's I'm glad you brought up monoclonals, because there the agency really, as you just pointed out, has been quite accepting to this concept. You have different targets, and it's partly because they know the human immune response is polyclonal and is based on many antibodies working together, and they seem to be very open to that. My understanding is that in the past they were less open to that, or at least quite demanding of you showing them a lot of data for combination products. And I think there still will be, to a certain extent, issues of toxicity or interference, like Miles mentioned. But in terms of saying, well, we want to have two drugs because there might be resistance to one and not to the other. I mean, in HIV therapy, that's total standard practice. It's total malpractice to use a single agent. So I think there's progress on that, but I think more discussion needs to be had, and more buy-in from the agency on how to do that in an efficient way.
Great, thank you. That's a great question. We have James Cummins put a question in the chat, and has going to hand it over to you, James. You can unmute yourself and elaborate.
Hi, Tom. Enjoyed your talk. So our contract in the Division of AIDS is the CRM contract that you referenced. And so to date, the only MPT that we've actually supported was MB66, so it was the monoclonal antibodies. So we have not yet been asked to support. I hope we do get asked to help provide some support for a contraceptive-based MPT, either hormonal or non-hormonal. So my question has to do with, are there any special considerations that we would have to consider to support such a product and the types of safety and toxicity assays?
Yeah, thank you, James, and thank you for speaking up the fact that program is available and perhaps underutilized, at least from our field. So I hope people will take notice of that and look at it. I think really it's really, I think where this is helpful is the pre-IND package, where you put forward a proposal. This is what we would like to do. These are you can be very detailed about it, and you can get FDA assessment of that, and then say, well, actually we want we want another arm, or we want you to extend the recovery period. They will be very specific if you're specific to them, and then that could guide, and you could move ahead in your program, knowing that this study will pass muster because it's been pre-assessed. And so if people are at the point of being able to assemble a pre-IND package, they can get that feedback quite quickly, and that would help you know that this study is going to be on target in terms of how it's designed.
Thank you, Tom.
Great. So I see that there is another question in here from Nanguri. Would you like to ask that, or else I can? It has to do with using dog and sheep as non-rodent animal models.
You can do it because the question is so simple.
Okay. Yeah, so this was actually to Tom. Yeah, the suitability of using dogs or sheep as non-rodent animal models, and just discuss, she'd like to
Right,
have you
right.
to discuss the similarity of these.
Yeah. So, you know, a dog is a very commonly used second species for products that need a second species, and that includes most things other than monoclonal antibodies, where a single species has generally been acceptable. So a dog is definitely one of the species, and the other are non-human primates, probably, correct me if I'm wrong, Miles, but I think cynomolgus monkeys would be one of the most commonly used ones. Sheep, less so, but sheep have got a special role in terms of vaginal rings and maybe even vaginal films, although those could be studied in primates or dogs as well. But the sheep vagina is large enough that a human-sized vaginal ring, in other words, your real product rather than some surrogate, can be used in the sheep. There are not that many providers who or contractors who do GLP sheep studies, but there are some, and sometimes that's a negotiation as to whether that study has to be GLP, but preferably it should be. And so finding a provider who can do that would be helpful. So in terms of the pro and con of the individual choice between primate versus dog versus sheep, a lot of things could go into that, including, you know, perhaps information about your API and what kind of toxicities might be seen there. But I think all three of those are options that could be explored. Miles, anything else you'd like to add to that?
No, that's pretty comprehensive.
Okay.
And you know, you can, for the device part of this, use a smaller version of your ring, for example. But this requires that you make a smaller version of the ring, which is, you know, can be a problem. If, for example, you wanted to go into cynomolgus for your
Right.
toxicity.
Yeah, and I worry a bit that whether at a GLP sort of definitive type study, it's not your product itself, and that's going to raise some questions as well. So certainly for efficacy, people have done that, and I think that that's been actually very valuable to do monkey efficacy studies, for example, with anti-HIV molecules. And I think people find that quite convincing. In terms of toxicity, it's often a good starting point, but I'm not sure it's part of the GLP type evaluation.
Thank you.
Thank you.
Great, thanks. So I have a question. So the FDA has guidance on exploratory IND studies. Can you tell us what this is intended really to describe?
Miles, would you take that one? You've been quite active in that area in terms of some of our studies.
Right. So, for example, you know, we have been working on a contraceptive antibody, and unfortunately, the target for the contraceptive antibody is restricted to male sperm, human sperm. And so doing proof of concept efficacy studies in other species was simply not possible. So in order to carry out a proof of concept of mechanism of action study, we filed an exploratory IND for the product known as ZB06, and then engaged or are presently doing a postcoital study to demonstrate the efficacy of ZB06 in contraception. Similarly, in addition to proof of concept mechanism action studies, exploratory INDs are also available when you're looking at PK, where you can't, where you're there's no good animal model for the PK of the drug, and you want to do this now in humans to test this. So it allows you to get into the clinic early to answer very specific questions about PK or mechanism of action with a much smaller or somewhat smaller non-clinical package. And then when you know whether or not your product has legs, then you would go back, do a more extensive non-clinical or non-clinical package and go into a standard safety phase 1. Tom?
Yeah, that's great. That's just what I'd hoped you'd cover. So I think it really can be helpful in a number of settings. There are other ways it's used, like for microdosing studies and things, which helps you get an early idea of PK and bioavailability. And that might also be useful for people with systemic drugs. That doesn't apply so much to the approach that we took, but there are other things you can do with an exploratory IND.
Great, thanks so much. I'm going to hand it over to Jeremy. You had a comment in here. I was hoping you could elaborate with the group.
Yes, thanks, Bethany. I was just going to go back to the discussion on products containing multiple APIs. So that there's three considerations for drug interactions that you need to address when developing such types of products. There's a pharmacological interaction, so where the APIs can interfere with their mechanism of action, as Tom referred to. There's the pharmacokinetic drug interaction, and there's the toxicological drug interaction. For the toxicological drug interaction, that would involve a combination toxicity study where you dose both drugs together and you compare them with drugs administered alone and a placebo or vehicle of some sort. The need to do those studies is addressed in the ICH M3 guideline, and it depends on the stage of development. So if you've got combinations of products that are already on the market, you may not actually need to do a combination toxicity study, or it may need to be done very late in the development process. Whereas if you've got two new APIs that have never been into a human, then you need to do the combination toxicity up front. So it's worth consulting the M3 guideline to get some more insight on that. And just to go back to the need to address the contribution of each API to the overall efficacy, the Code of Federal Regulations actually requires you to do so. How you do that is not addressed in the CFR. Typically what's done in pharmaceutical development is that's addressed clinically, but for combination products for prevention, that may not be a practical approach. And I don't think anyone's actually addressed that yet with regulators to come up with something that is feasible. But you do need to demonstrate that any component, active component of a combination product does contribute somehow to the overall efficacy.
Yeah, just to elaborate on that, obviously for antiviral antibodies, essentially and we were alluding to this earlier, the agency has given that something of a pass, in the sense that if you can show in vitro, say, a different range of neutralizations for variants which are circulating, that essentially or at least has, so in the COVID field and so forth, or HIV field, been sufficient to justify the presence of both antibodies.
Yeah, I should have specified I was referring to drugs, not biologics or anything else. So yeah, that's true.
Yeah, Jeremy, thank you very much for those comments. I think that's really helpful. And since a lot of the field is looking at these kind of combinations, that's really a question people are actively facing. So thanks so much for weighing in with those comments.
I want to just say that Miles and Tom have graciously offered to stay on an extra 15 minutes past if we have more questions. And I see Joe is raising his actual hand for questions.
Yeah, Jeremy, just to follow up on the points you made about those required studies, do they all necessarily have to be done with the actual product, or can you do them by infusion experiments or other alternatives to the actual formulation when you're looking at?
No, it doesn't have to be with the actual formulation. In fact, in most cases, that wouldn't be practical. It's the interaction of the APIs that the regulators are concerned about. So you just have to find a way of delivering both APIs simultaneously.
So delivering them at least at the level you're expecting to have with regard to the actual product in the end.
Higher. You need to be able to demonstrate
have a margin, right? Okay.
A margin of safety. So you need to go higher than the clinical dose. Yeah.
Thanks.
Good. Thanks. I see that Leslie Marshall had a comment. Do you want to elaborate on that, Leslie?
Sure. I'm a core working under James Cummins' crimp contract. And I just wanted to share that we have supported IVRs and utilized the sheep model for safety assessments through our contract with ABL. And these GLP studies were actually completed at Sinclair Church. And I've included the link to their website in the chat box.
Thanks a lot.
Thank you. That's an important resource.
We did get some questions submitted ahead of the webinar from many of you, and Tom and Miles have them and have looked them over. So Tom, do you want to address any of those?
I'd like to bring up and maybe and comment on it. How to assess toxicity in vaginal microbiome, as well as, how to assess toxicity to the vaginal microbiome, and how these increase STI susceptibility and toxicity of delivery systems and the materials they're made from. And I don't know, I'm hoping that Jacques might comment. The obvious thing is we certainly feel we want to support a vaginal, lactobacillus dominated flora. And if we're not doing that, or if we're in fact in the opposite, moving toward vaginal dysbiosis, I think most of us are pretty concerned that that would be bad. But I think there are some finer points there that perhaps Jacques could comment on.
Thanks, Tom. This is Jacques Revel. Yeah, I agree. I mean, the broad definition of lactobacillus, no lactobacillus can be applied, but I think that nowadays, if you're serious about your product and you want to bring that to FDA, you need to show some more molecular data to establish the composition of the microbiota pre and post use of your product. The big challenge is obviously when you start moving to animal studies. I mean, I'm one believer that it's not representative of what's happening in human. So really that the effect on the composition of the microbiota can really only be established in human.
Thank you, Jeff. And then another one that I got was, what is tolerable toxicity? And so that sort of brings up, there's a term of art used, they talk about safety and tolerability studies. And so, you may, for a given drug, you may have adverse effects, but they're tolerable. So, you're still able to go forward. And I think that's true for these kind of products as well, but the tolerable level is lower, as we talked about in one of the first slides, because you're starting off with people who are healthy and they're using this to try to stay healthy. Their tolerability for side effects is less. Now, and still with the goal, particularly for contraception, women with hormonal contraceptives tolerate some significant, everyday nuisance side effects. And they still find that tolerable because what they're getting is very important to them. And so I don't think we should be too shy of feeling that some side effects of these drugs can be tolerable and still be a successful product, but it is less than certain other products that are used to treat ongoing conditions. So I guess that's what I'd say as what's tolerable. I guess one thing that's not tolerable, I think, is if your product actually damages the epithelial barrier or does other things like that might enhance transmission of STIs. That's something of great concern and might make it not approvable or might make consumers very unlikely to choose it. I think those were the topics that were brought up in that group of questions that I thought would be of interest to the group. So thank you, Bethany.
Great, thanks a lot. So are there any other questions from the group? If so, you can unmute yourself and pose them directly. Otherwise, we are at the top of the hour. As I said, we could continue going on, but we would really like to thank you all again for participating in this webinar. and especially to Miles and Tom for your preparation, Tom, for your great slides and for this great discussion. So we will be having the recording on the website, and we'll also circulate it around to everyone in a couple days. And with that, I think that's it. And we just really want to thank everyone again, and also to want to acknowledge NICHD and USAID for supporting this work in opposite AIDS research. So thanks a lot, and we'll see you soon. Bye-bye.
Thanks, Bethany. Bye.